All right. Got that over with. All right, y'all. I'm Monty Bain, the Southeast Regional Communications Manager. And as y'all know, I cover Florida to Virginia. And this meeting is about North Carolina, South Carolina, and Virginia. And if you want to go to the next slide, Stacy. All right, this is a little bit about what the Cotton Research and Promotion does. The Cotton Board is overseen by the USDA, and we collect the assessments, we administer the program, and we communicate to the stakeholders. And one of the ways we do it is through these Zooms, now that we've learned to do it through COVID. And of course, the main way is our tours. And Cotton Incorporated conducts the research, promotes cotton to consumers, and creates demand and profitability. And thank you, Cotton Incorporated, for getting us dollar cotton. The assessments are producer, and in the 90s, we also added importer assessments. The producers pay about 53% and importers 47%, and we're over 99.9, I think, compliance rate. Our budget for 2022 is $82 million, and that's up $2 million from 2021. The Cotton Incorporated State Support System brings back 7.5% of assessments collected in each state. So South Carolina, for instance, their assessment, 7.5% goes back to their local research projects. And funding is determined by a committee in each state, and that's made up of Cotton Board, Cotton Incorporated board members, research, and CPOs. And the research projects are managed by Cotton Incorporated's research staff. These are the state support committee members in North Carolina, South Carolina, and Virginia. And I attended the South Carolina meeting a couple of weeks ago, and they were listening to their research projects. But we want to list all of these. Virginia usually has theirs in late August, and North Carolina the same. And with that, I would like to go ahead and turn this part of the meeting over to Ed Barnes, the Senior Director of Ag Research at Cotton Incorporated. Ed, you're muted.
You know, someday I'll get that figured out after about 7 years of Zoom, I think. And hopefully I did share my screen now.
Yes.
All right. Well, I appreciate that, Monty. And I'm just going to go into a little more detail on what Monty talked about. First of all is to talk about how the funding works. And, you know, the good thing is it's based on a 5-year Olympic average. And so what they do is they look at the bales produced over the last 5 years and throw out the high and low years and average the middle 3. And so we just finally got rid of 2015 and 2016, which were not great years. We had low acres, and many of you will remember some of the hurricanes from that time. So I'm glad that's out of the past. And you know, we had some challenges in 2022, so that's now our low year. And we had kind of a good year last year. So our state funding levels now are headed up a little bit, and you can see how each state, the funding level is proportional to the number of bales in that chart. The light gray bar is Virginia, the orange is South Carolina, and the blue is North Carolina. And so this is just that 7.5% that Monty talked about that goes back from our national assessment. And it doesn't include any funding that comes from our core program. I'll explain that a little more in the next slide, but that is where we have national and regional focus projects, and that's managed out of our carry office. And then we're really fortunate in the Southeast that all the Southeast states have a very strong state checkoff. And so this does not include money that comes from the Virginia Cotton Board that we coordinate with Gail Miltier in Virginia. In North Carolina, the North Carolina cotton producers have their own checkoff, and we work closely with David Parrish. And then finally in South Carolina, They have a cotton board as well, and Catherine Helms is with the South Carolina Cotton Board. So we work with all these individuals to make sure that our Cotton Incorporated funding is not duplicating anything that's being funded by the state, and it's all well coordinated. One of the ways we do that also is through our ag research staff. And most of us here kind of wear two hats. We have an administrative hat where we help serve as liaisons between Cotton Incorporated and the state support committees. And so there you can see in the top left corner who covers the different regions. I share the Southeast with Dr. Don Jones. Don covers Florida, Alabama, and Georgia. And then of course I do Virginia, North Carolina, and South Carolina. And then above that is we all have our technical expertise. And so I'm going to show you later a list of projects that's funded by each state. And in this process, when the researchers submit proposals, they come here as well. And I have all the staff here. If it's in their area, for example, if it's something on controlling weeds, then Dr. Galen Morgan, there shown at the top right, would review that and give me any technical comments. And ultimately, though, for the state support projects, it's the grower committee that selects what's important to them. And we just provide if there's any technical questions, we provide some advising. But once those projects are selected and funded, then it's up to us. If something in weed management is funded, then Galen will manage that project and make sure it's coordinated with other states and his overall core program. And so at Cotton Incorporated, I'm excited. We've got a couple new faces on this page now. And so most of you know, some of us that have been around, Dr. Cater Hake is our vice president, and he's been covering disease and the plant pathology that's been vacant. But I'm very excited. There you'll see under the word disease is Dr. Caitlin Bissonette. She is the plant pathologist for the University of Missouri and Extension. And she has accepted that position and will begin on April 4th. So we're very excited to have her joining our team. I mentioned Galen, who does weeds and soil health. He's been around now for 3 years, which is kind of hard to believe. We're lucky to get him from Texas. I'm an agricultural engineer, so I oversee harvest and ginning and some of the automation projects. Dr. Brian Kurtz is our entomologist, and I'll say some more. He also helps with this grower outreach we're going to talk about in a second. Tom Wiedegartner oversees cottonseed. And then another new addition, we're very excited. She just started last year is Dr. Susan Jaconis. She goes by Evie. We stole her away from kind of the lentil board, what does some different promotion. And so she has a good background in human nutrition, is going to help us promote cottonseed oil. And then last but not least, of course, Dr. Don Jones, who's been on staff for some time, overseeing our breeding and genetics program. So each of us, once these projects are funded, we work, and just make sure that those projects are being executed as planned. This year, I'm going to go through what we're going to do. Is last year we kind of highlighted the projects that were funded in North Carolina. This year we're going to highlight South Carolina, but just to give you a quick list of, I'm not going to read through these. I'm going to let you look. And if this is, watch the recording later, you can pause here to get a better look at what's funded this year in North Carolina. Last year we had Dr. Guy Collins present on activities of the North Carolina Support Program, and you'll see Dr. Collins is not listed here. That's because he is funded directly by the North Carolina cotton producers. And so, again, an example of how that is coordinated with our state support program. When we look to Virginia, I know Dr. Hunter Frame is on this call, and he is the cotton specialist in Virginia. And you can see the projects that are funded by Virginia. Again, the ones from the Virginia Cotton Board are not shown here. And then finally, South Carolina. And when you hear Dr. Jones present, you'll hear a lot more projects than those listed here, because he's going to include things that are funded by the South Carolina Cotton Board as well. That's going to conclude my comments. Before I introduce Mike, I just wanted to call out 2 online resources. We do have on our website a listing of state support projects. That top QR code would take you to the link that's shown there. And then another thing I just want to keep reminding people of, because I'm very excited by our cottoncultivated.cottoninc.com. Dr. Ryan Kurtz, I mentioned, has been leading our efforts in electronic outreach. And that website was just redesigned. Ryan did a wonderful job. And so he is going to, and I know Monty's going to call this out again at the end of the webcast, but just to make another plug, on March 15th, Dr. Kurtz is going to present a Cotton and Coffee, where he's going to kind of go a deep dive on all the features of this new website. I'll call out two things just to maybe try to inspire you to go there if you haven't already. Right when you hit the landing page, there's going to be a video that is going to be, it's updated about every Thursday or Friday on the weekly outlook for weather forecast from Eric Snodgrass. And most people I talk to really find that informative and helping plan out, what to expect in the coming week. And then one thing that's a little tricky, and if you scroll down from that page, you'll start seeing the latest webcast. And that's another thing Ryan has worked on with the Plant Health Network. Just some really great webcasts that are available on demand that allow us to cover really current and timely topics. And so with that, I'm going to conclude my comments, and I want to introduce, and I really appreciate Dr. Mike Jones. Mike is a longtime cotton specialist at Clemson. Mike, I think as long as I've been around, and that's 19 years, I think you've been the cotton specialist there in South Carolina. And
yeah, I've been here 23 years now.
23 years. So Mike obviously has tremendous experience, and I really appreciate, and you're going to see that he is well informed of not just his own program, but everything cotton in South Carolina, and really helps keep all that tied together. So with that, I would be glad to turn it over to you, Mike, and really appreciate you joining us today.
All right, if I can find my, there it is, I think. All right, can you guys see that okay?
Yep, that
Looks
looks
good.
good.
All right, as Ed mentioned, my name is Mike Jones, and I'm the Extension Cotton Specialist with Clemson University. I am going to give you guys a very quick, brief summary of a lot of the research projects that we've done. I'm not going to talk about all of them because there's just not enough time to do that. And I'm going to move through them fairly quickly. And if you have questions, you can call me later or call the investigator in charge of that project and ask them some questions about it. Before I do that, I want to thank the South Carolina Cotton Board and Cotton Incorporated for all the work that they do for the South Carolina cotton farmer, Clemson University, and the researchers at Clemson. These, of course, are grower-funded organizations, so we want to thank the grower too, because we're working for them and we appreciate the funding. All right, so the first one I want to talk about, the first project that was funded is entitled Evaluation of Elite Conventional Cotton Breeding Lines in South Carolina. The PI on this is Dr. Todd Campbell, and Todd is a talented research geneticist. He's located here in Florence, and we're lucky to have him. He is with the USDA ARS. His research basically uses traditional breeding methods, genetics, genomics, and high throughput phenotyping to try and improve. His main topic is fiber quality. So one of his main goals is to breed cotton varieties that have really high fiber quality. But another priority in his program is to make sure we are maintaining the genetic base and increasing the genetic diversity of our cotton lines, which is sometimes can get kind of narrow. More along lines with this study, though, is he wanted to take some of those elite lines with high fiber quality and try to evaluate those for fiber yield, so that some of these varieties might be used by industry in our area. So the objective of this project was to evaluate the yield and fiber quality of elite conventional germplasm that Todd has developed over the last few years. We conducted these trials in Edisto, which, as you see the map on the right, is the one further south. That's in Blackville, South Carolina. I had a location for Todd in Florence, and he actually went across state lines in Jackson Springs, which is a real sandy, droughty soil, and planted and evaluated these varieties there. And really what he's trying to do is not really commercialize a conventional cultivar, although he could do that if growers were interested in that. but he wants to release them and have them used as breeding parents, so companies can develop varieties that are adapted to our area, and they can put transgenic traits in those. And here is a little bit of the data. This is the data from Florence and the irrigated and dryland trial in Jackson Springs. Thing I want you to point out is I highlighted Delta Pine 1646 in blue. That is the standard variety for the Southeast. It's probably on 50 to 60% of the acres. A real high yielding commercial variety, transgenic variety with a good fiber package. If you look at the seed cotton yield and lint yield data that Todd has developed with these new strains, you can see 6 or 7 of them are equal or better than Delta Pine 1646. And I think that's extremely important because a lot of the varieties that we evaluate in our cotton trials, our variety trials, commercially released varieties don't yield with 1646. The lint percent, you can see most of his varieties have less lint percent than 1646, and they're smaller bolls. 1646 has smaller bolls than a lot of the PD lines. So they're getting their yield from different places. Again, Todd is really prioritized fiber quality, and if you look at the length and strength of his varieties compared to 1646, you can see they're way beyond what we consider to be a really good commercial variety. So in summary, 6 of his lines were equal and are better than 1646 in yield. 5 of them had fiber lengths that were only 5 to 7% lower than what's required to be Pima cotton. So some extremely high quality cotton. 3 of the lines produce fiber strengths that meet the requirement for Pima. So I think that's real exciting. He's going to release 4 of those this year, and we're going to keep evaluating some of his newer material. The next project is engineering insect resilient cotton. This is a project that's just in its beginning phase. Dr. Chris Saskey, who is an associate professor of systems genetics, is the PI on this project, and he's up at campus in the Department of Plant and Environmental Sciences. This is, again, maybe a little bit beyond what I can help you guys with. So if you have questions that go beyond what I tell you, you can call Dr. Saskey. But right now, you know, most of the cotton that we grow is transgenic cotton. It's BT cotton, where it has proteins that are inserted into it to give it insect control. The first BT cotton came out in 1996 and was really good on worms. And then following generation, 2nd and 3rd generation BT cottons were released that improved the effectiveness of those proteins in cotton on worm control. But again, most of them are only effective against bollworms, armyworms, and soybean loopers. And you can see sometimes, depending on the time of year and the population, we have to overspray those with insecticides. So there is a chance that we could get some resistance to the BT technology. We do have other insects that attack cotton, such as stink bug, spider mites, thrips, and aphids. So Dr. Saskey is trying to come up with a novel approach to control these pests and nematodes. So what he's done is taken the genes out of sorghum that can actually produce a toxin called hydrogen cyanide. So the insects do not like this toxin. It's naturally occurring in the plant, and it really gives you broad spectrum insect and nematode tolerance. So there's not a lot of pests that attack sorghum, and the reason for this is that it has these genes for cyanogenic glucosides that convert into hydrogen cyanide. They've even shown in Arabidopsis that this gives tolerance to beetles and stink bugs. So that's pretty interesting. Again, he's in the initial phases of this project. So what he's trying to do is synthesize the 3 genes that give the tolerance to these pests, and then transform cotton by placing them in a plasmid, and then transform, making a transgenic cotton, and then selfing those plants, put him in cell and tissue culture and selfing those plants until he has a transgenic cotton that has naturally occurring resistance. He did meet some challenges this year. He couldn't get one of the genes to work like it was supposed to and get it into the cotton plant. He was able to figure out how to do this by making the pieces smaller, and he's going to try to do that this winter and get that project started. Next is Dr. Eric Billman's project, and it's entitled Perennial Ground Cover Crops in South Carolina Production Systems. Dr. Billman is a new research agronomist with USDA that took Phil Bauer's place when he retired. This is the first year for this project, and Eric's specialty is cover crops. So instead of using annual cover crops, which is normally used in cotton production systems here in South Carolina, Dr. Billman wanted to look at establishing perennial ground covers and leaving them in the field all year and planting cotton into those. The species that he wanted to look at were red and white clover. He wanted to look at vetches and annual grasses, fescue and annual ryegrass, perennial ryegrass. Again, he's thinking that we can reduce the herbicide use of our management systems by doing this, conserve water, and decrease our insect populations. So again, you can read through his objectives. I've just covered that. And he wants to try to do this without decreasing the yield or the fiber quality of the cotton crop. This is a little bit of the data from this year. He's working with Mike Marshall, who's our weed scientist at Edisto, and measuring weed pressure. And the bar that's really important is the brown bar. That's fallow. That's where we'd have no ground cover. And you can see where he added the clovers. We've got significant reductions in all weed pressure. Almost eliminated all the broadleaf weeds out there. Some of the grasses snuck through, such as goosegrass and that sort of thing. But it did a pretty good job of suppressing weeds. And then he also got Dr. Francis Ray Jones to come in and measure some insects in these plots. And the biggest thing was he was able to reduce his early season thrip pressure by growing these clovers with cotton during the season. All right, so now I want to talk about a different kind of project. Dr. John Park is a senior scientist located here at Florence at the PD Research Station. He is a molecular geneticist and breeder, and he works in cotton and oilseed brassica plots. This is a little different kind of work. It's interesting, but basically what he's trying to do is chemically induce mutations in cotton genes to increase the genetic diversity of cotton and try to increase yield and quality at the same time by using these mutations. So his objectives were to evaluate selected TTU lines, which is Texas Tech lines that have already been mutated, evaluate those in South Carolina, and then he wanted to mutate 2 lines, an Arkansas line and a PD line, with this chemical to see if he can increase fiber quality and yield. And then he's also planning to do that in some Pima lines that we have here on the station. And this is just a slide showing the treatment. First, you have to figure out how much of this chemical ethyl bethane sulfonate that you can use to cause the mutation and still get the seeds to germinate. So he treated 10,000 seed with a 2% solution of this EMS chemical, and then planted those out in the field. And then every year of funding for this project, he's been out there doing selfing and an increase in the seed population. So he's now to the point where he's evaluating these varieties for yield and quality. This is a little bit of his data, and it's real busy, and I'm not going to go through all of it. But the first slide just shows he got a 10% increase in fiber length and strength from these mutations compared to the parents, which are the arrows. He's able to increase bowl size and seed size in a lot of these lines, and he increased the protein in the oil of some of these lines by doing this mutation. All right, now we're going to get from a real basic project to one of our extension agent projects. He got some funding from the South Carolina Cotton Board, and he wanted to evaluate the water quality of what we're putting in our spray tanks in South Carolina. We know water is very important in our spray solutions. It's almost all of 95 to 99% of what we put in the tank. And we live in sandy coastal, most of our cotton is grown in sandy coastal plain areas. We either have shallow wells that are really good quality, but not a lot of water, or we have deep wells that are have very poor quality, and this can affect the activity of the chemicals that we spray. So this year, William was able to sample get 98 samples from 23 counties, and you can see those in the orange, and he sent them to the Clemson's Ag Service Lab to get them analyzed. And the biggest thing that he found was we have issues with pH. Most herbicides work really good between pH 4 and 6. There's a few that don't, they like more alkaline. But as the pH increases, the spray solution, the longer you leave it in the tank, the less effective the chemical is in the spray solutions. So this is a busy slide. I'm going to touch on it real quick. The first bullet, he found a wide range of results in the testing area, even within the same county. So really, we need to be testing every water source that we have. Hardness ranged, had a big range, but 26% of those samples were over 100 parts per million, which concerns me because that can reduce the effectiveness of a lot of the herbicides that we spray, such as Roundup and dicamba and Post. You see the range of pH was from 4.3 to 9.1, and a lot of those 66% were over 6.5, and that's when we start having issues with our pesticides. 11% had high salinity and 13% had iron issues. And these are important because they can clog up your sprayer when they come out of solution in your sprayer tips. So, all right. This is an interesting project, and this is one that I think Ed picked up as a core project from a assistant professor of crop physiology that we have on campus. Her name is Shruti Narayan. And I'm helping her with this project, and Dr. Todd Campbell and Dr. Kara Parthi are providing variety germplasm for her to do this project. But she really wants to take a look at the roots of cotton. Roots are extremely hard to measure. They're labor intensive. They're hard to measure in the field. So she's come up with a system to do root measurements in the greenhouse. And she wants to look at the variability in these different genetic bases of cotton to eventually try to come up with some way to molecular map roots and their growth characteristics, which would help our breeders use these in breeding programs. So she was able to get her hands on this upland core set which really has about 90% of the phenotypic differences in cotton over the last 100 years. And she picked out 20 of those and grew them in the greenhouse, and she grew them in these little PVC pipes on the left. She calls them mesocosms, but what they are just PVC pipe that are 3 to 4 foot long. She puts this product in them called turfus, which is a clay material that's really, really similar to the coastal plain sands that we have here. And then in half of those, she actually put in a paraffin wax plug about 15 inches into these PVC pipes to simulate a hard pan. And then after the cotton matured, of course, she washed the roots and measured the length and the amount of root material that's there and took pictures of them. Very interesting work. One of the interesting things that she found was there was a considerable amount of variability out there, which is good for breeders, especially when we're trying to breed for drought tolerance. Of course, the roots are going to be very important there. But some of the actual genotypes were able to penetrate a hard pan, which I thought was really interesting. So she's going to continue looking at these varieties, genotypes, and evaluating the roots. Okay, now this is getting really basic. We have a assistant professor of molecular breeding and a faculty scholar at Clemson named Dr. Sachin Rusky. When Dr. Rusky started a few years ago, he asked me what some of the problems were with cotton. And I said, the biggest headache we have, Dr. Rusky, is that cotton is a perennial, and it doesn't really know that it's going to be cultivated as an annual. Why being a perennial, why is that important? Well, cotton thinks that it's going to live through the winter. It doesn't know that we're going to defoliate it and that cold's going to kill it. So it saves quite a bit of carbohydrate and nutrients to regrow. And that's why we have regrowth problems sometimes also. Cotton does produce a lot more fruiting structures during the year than it can retain because of this. In a good year, cotton normally will kick off about 50% of those fruit. Sets a high percentage of them early when the carbohydrate supply is high, but as the boll load increases, that plant wants to live to the next year, so it kicks a lot of those fruit off. So if we could tap into some way of tapping into those reserves, we could probably increase our yield significantly. I was surprised when I mentioned this to him that he said he probably could work on that. And I asked him why, and he said in a plant species called Arabidopsis, they've been able to isolate 5 genes that actually control the annual and the perennial and indeterminate determinate growth habit of that plant. If you see that last bullet, there's an SOCI gene and an FUL gene, and they control flowering time. In Arabidopsis, there's an FT gene that controls the balance between being an indeterminate and a determinate plant. And then within that same pathway, there's a LFY gene and an API gene that control the meristematic activity. So he went out to try to isolate this in cotton, and he actually found those genes were highly active in cotton. He found 45 different expression traits in cotton for these 5 genes. They were really high in 8 varieties, and that's what he's shown on the bottom left in that table. Those 8 had high expression levels of these genes, which made them actually a little more determinate, a little more annual. You can see none of those total points added up to 100%. So with that being said, he's tried to take these 8 varieties and he crossed them with each other to try to stack more expression into these. So he's made 561 crosses of these 8 varieties, and he's growing those out in the field and in Costa Rica. I'm not going to get into this slide other than he was able to find a lot of variability using the 63K SNP chip in cotton, and 23% of them had a polymorphism, which is very important when you're working with cotton. And then he was able to actually isolate those 5 genes on the chromosomes of cotton. And so he's he's trying to again stack those on the chromosomes now and get a little more effective expression out of those genes. And really, he's proved that he could do this. This is a busy slide, but on the left, he's injected a virus into the cotton plant, which blocks the pathway of that AP1 meristem gene. And what happened there is where he blocked that pathway, he got a normal fruit, and where he increased the amount of that gene, he got a 4th bract to develop. So again, it's very basic right now, but he's trying to tap into the perennial reserves of cotton. This next project is host status of new cotton varieties to various nematode species, and the PI on this is Dr. John Mueller. Dr. Mueller is a professor of plant pathology, but actually works mainly in nematodes, and he's located at the Edisto Rec. As you know, over the last few years, 2 companies, Phytogen and Delta Pine, have released nematode-resistant varieties, and they really have very little yield drag compared to varieties that don't have this resistance. And he's listed those here for you. There's 6 that only have southern root knot, and that's the SRK resistance. And then there's 2 Phytogen varieties, 443 and 332, that also have reniform resistance. And then Delta Pine has 2141 on the market now, and it has southern root knot and reniform resistance. John is concerned that growers who will plant these resistant varieties to root knot and reniform will forget that there is another nematode out there that can impact yield of cotton severely. So this study is looking at Columbia lance nematode, and this is a survey they did in the early 90s. It was published in Plant Disease, where they surveyed as many fields as they could in South Carolina and found 61% of those fields had Columbia lance nematode in them, and 37% of them were above treatable levels, which means it would cause us a significant yielding decrease. So John looked at a study, and first of all, he wanted to create an environment where he didn't have any nematodes. So he put 3 gallons of Telone out, which is very effective against all nematode species, and in a low pressure field of Columbia lance and in a high pressure field. And he was able to show that there were Columbia lance nematodes in those fields by doing this. And then he planted the 6 varieties and evaluated those. And basically, he didn't find any effect of the southern root knot resistance or the reniform resistance on Columbia lance nematode. So it is going to be a problem in our fields. And then this is a large plot test that he did. And then the last trial I want to talk about, a project I want to talk about, is evaluation of cotton insecticide wash-off intervals for pesticides. And this is, PI on this was Michael Plumley. Michael Plumley was an assistant professor at Clemson working in cotton, but now he has actually been switched over to be our corn and soybean extension specialist. And he worked with Dr. Jeremy Green, an entomologist at Edisto, on this project. And basically, they wanted to know how long insecticides would be effective on cotton after it was sprayed. A lot of times we'll spray cotton and it'll start raining while we're in the field or 30 minutes after the field. And of course, we don't know if we need to respray these or not. He did 3 separate fields at the Blackville Edisto Rec location, and he was targeting 3 major insects, thrips. So he was spraying acephate to control thrips, Prevathon to control bollworm, and that was non-BT conventional cotton, and then bifenthrin to control stink bugs. And then they came in with they sprayed, and then they came in 30 minutes later with just water in the tank and washed that off after immediately, 0 to 30 minutes, 1 to 1.5 hours, 3 to 4 hours, 6 to 7 hours, and then 24 hours. And Then went in and sampled those. Unfortunately, they didn't have very good thrips pressure this past season. It's good for the grower, but bad for these guys doing the research. So they didn't find any differences in thrips, and they didn't find any differences with the bollworm, which showed that it's pretty effective on bollworms. No matter, as long as you get it out there, it stays on the plant pretty good. The only differences they really found were with stink bugs. So if you get a rainfall within 30 minutes after you spray for stink bugs, you're probably going to need to respray those fields. All right, and this is my last slide. I know I'm going a little long. We have other projects that were funded. We have a soil fertility specialist, Bhupender Farmaha, and he was funded with 2 projects, and he really wanted to look at the cotton recommendations that we have in our growers guide and see if they are still valid. So he went to on-farm locations and looking at different nitrogen and potassium rates. Dr. Jose Pereiro is our irrigation, extension irrigation specialist, and he is working on developing a cheap automated wireless soil moisture system to control your irrigation systems. There are more commercially available ones on the market, but he's trying to do this very, very cheaply. I appreciate the funding for my 3 projects, and I'm not going to talk about those, but I was able to get funding to expand our OVT program off the research stations and onto growers' farms. And I think that's improved our results that we're getting from our variety trials. I also get a lot of questions about Sea Island and Pima cotton, why we don't grow that here in South Carolina, and basically it's because we don't have varieties that are adapted for our area. But we're looking at some Pima and Sea Island cotton with Dr. Todd Campbell and starting to breed a little bit for that and see if that might be a viable option for us. And then we also get support for our South Carolina State Cotton Meeting. We had our 20th annual meeting this past January. It was well attended, had really good speakers, and I appreciate the funding for that. Finally, Dr. Mike Marshall is our weed scientist. He's surveying for Palmer amaranth resistance in South Carolina. Fortunately, we do not have any of that other than Roundup. And then Michael Plumley got 2 more projects that were funded by the Cotton Board, and he is looking to evaluate cotton module traceability and quality mapping. And basically what that is he's using that RFID that Ed talked about, and like a yield monitor in a cotton picker, and he's actually, instead of doing the yield maps, he's doing that based on fiber quality. And then he's also looking at seed size and seed quality in the final project. So that's all I wanted to share with you. Again, if you have any questions on any of these projects, be glad to give me a call and I'll discuss them with you. Or you can call these investigators, these scientists yourself.